Good morning, everyone. On the first event of the institution. This is a one-day workshop on entrepreneurship and innovation as career opportunity. I'm delighted to have you all here today, and I would like to extend a warm welcome to the speakers, our director, Dr. Mishra, who is also the president of the IIC IRSHA, members of IIC IRSHA, faculty, students, and staff. So I will just introduce Institution Innovation Council. The Institutional Innovation Council actually is an initiative by the Ministry of Education through the Ministry of Education Innovation Council. This program of Institutional Innovation Council has been launched and for research in higher education in the year of 2018. And the program is basically designed to culture of innovation and startup ecosystem in higher educational institutes. So the primary role of IIC is actually to engage the faculty, students, and staff in various innovation and entrepreneurship related activities like ideation, proof of concept, problem solving, design thinking, or intellectual property rights, project handling and development at a pre-incubation or incubation stage. So today we will embark on a journey of discovery, innovation, and learning. And our distinguished speakers have a wealth of knowledge to share with us. I welcome them again and believe that their expertise and insights would be invaluable to our audience, which basically consists of PhD students and scientists. So I would now like to request our director, Dr. AC Mishra, to share a few words.
I think you are going to have an inauguration somewhere
Before lunch, sir.
before lunch. I welcome Deshpande sir because he is very renowned biotechnologist. So we first go through the talk what he is doing and then during inauguration time further formalities facilitating the. So sir, I request.
So we have our first session. Our first speaker is Dr. Mukund Deshpande. He is the former chief scientist of CSIR NCL and director of Green Invention Biotech Private Limited. So just to introduce him, Dr. Mukund Deshpande is a distinguished scientist and educator with a rich academic and research background in the field of microbiology and biotechnology. With MSc in microbiology and PhD in biochemistry, he has achieved academic recognition with Doctor of Science degree in microbiology, which has in the 1994 by University of Pune. His thesis was on fungal differentiation and biochemical correlates, which has marked a significant milestone in the field of fungal biology. And throughout his illustrious career, Dr. Deshpande has been a prolific researcher, successfully completing over 35 research projects. His collaborative work includes prominent programs such as Indo-Swiss collaboration in biotechnology and the Indo-Belarus program showcasing his ability to engage global scientific endeavors. Dr. Deshpande's excellence in his field has recognized through his fellowship in the Maharashtra Academy of Sciences in 1994 and Society for Biomedical Advancement since 2010. His research contributions are extensive, comprising of over 160 research papers, reviews, patents, and authoring 12 book chapters that serves as a vital reference in microbiology and biotechnology. As an educator, Dr. Deshpande has guided and mentored 21 students to successfully complete their PhD degrees, leaving a lasting impact on the next generation of scientists. His entrepreneurship spirit led to the establishment of Green Invention Biotech, a startup focused on agricultural biotechnology, where he developed and licensed three groundbreaking technologies for pest and pathogen control. Beyond academia and entrepreneurship, Dr. Deshpande is also dedicated to science outreach and engagement. He has organized the 50th Annual Conference of Association of Microbiologists of India, that is AMI, and conceived the film Lokmanya Tilak, The Unsung Scientist, to bridge the gap between scientific research and public understanding. Furthermore, his spin, technology for production and micro-insecticide using spores of Metarhizium aniscolia, he showcased the application of technology in agriculture and contributed to scientific knowledge dissemination. So, in summary, Dr. Deshpande's career is an evidence of his profound contribution to microbiology and biotechnology, his commitment to education and mentorship, and his dedication to sharing knowledge with the world. His impact on the scientific community and society at large continues to inspire and shape the future of these fields. Thank you, sir. I now welcome you to please address the crowd. We have a second slide changer. He wants is it
Good morning, everybody. Actually, whenever I hear this introduction, it gives me a lot of pressure because the expectations are more. I don't know whether I will fulfill those expectations, but I will try to fulfill your expectations. Actually, I am waiting for the slide changer because it is very difficult to tell and change the slide by telling that okay, please have next slide, and it is obviously creating problem for him also and for me also. Actually, what I want to say that I want to share my experience and how I started this company with my one of the postdocs, Dr. Santosh Tupe. And while talking about my experience as a case study, I would like to have some information about entrepreneurship to you and how the title of my talk is the Discover Your True North. It is not that always easy to have, okay, this is my thing, and this is my process, this is my product. It is very difficult to understand yourself on this aspect. So I thought that maybe this will give you idea how one can go ahead with the entrepreneurship. I was in CSR for almost 43 years and I retired as chief scientist. The purpose of this laboratory is actually that the advanced knowledge and the applied chemical science for the good of the people. When it is written there, and it is for National Chemical Laboratory because for chemical science, there is National Physical Lab, there are other labs also, where the purpose is same that you should work for the people. But does it really happen? So that is the main question. Okay. Now, the first thing which I would like to make it very clear that number of times research institutes to scientists they say that okay we are basic scientists. It is not the case. There is no basic or applied science. Can you have this pointer? Yes, he's doing yes. Okay. Okay. There is no basic or applied science. Only the time frame is different. Whatever science you are doing, only the time frame is different. There you can use that knowledge for the application. Now, that's why I mentioned that it is a concept research and the knowledge-based application research. So I would like to divide this basic science and applied science into two aspects. One is the concept research, which usually we do carry out in the research institute, and the same thing. Though people do not agree, but R&D departments of most of the industries they also carry out concept research as well. Now, so now in the research institute, you will find that the performance is adjudged by publications, patents, projects, popularization of science, and finally paisa. But this is the five P's which our earlier director Dr. Ratna Swami used to say. And these are the criteria for the promotion or for the APAR, etc. Now, how many people read your publication? Four? Yourself, maybe as you have written, editor, two reviewers, and maximum 200 if it is worth reading. What is about patents? How many people use your patent? Only 1% of the patents are being used. So that means most of the time it is none. What is the outcome of your research project? Only the completion report, because at the last date is coming closer. We only work for the project to distribute. How much time your popular lecture or article has impact? Not more than 24 hours. If you write something for newspaper, next day you know where that paper goes. How much paisa you brought from government? Number of times all research institutes they get money from the government. So substantial money you can bring. Even I also could bring lot of money from government. Industry practically nil, and that is the major gap that industry academia interaction is important. Still, you get upper excellence, but on the other hand, how many people can use your product or process, whatever you have developed or whatever you have done in your institute? Thousands or more. If you do this one, certainly you will get a better recognition. The Indian biotechnology industry holds only 2% of the share of the global biotech industry. That means we are really not doing much biotechnology per se. Now, our government always has a dream that we should have 100 billion US dollar industry by 2025. Every year we are increasing. Actually, earlier it was 2024. Now we are saying that it is 2025. So every year we are just going ahead, but not we are not really getting that much amount of the biotech industry in India per se. The government has to invest minimum $5 billion US dollars to develop human capital, infrastructure, etc. So I personally feel that this is the job of the government, and then only the students or the entrepreneurs can work really hard to get the different types of industries. Now, this is the general sequence in biotechnology, and now you have to identify yourself where you can fit yourself. Application objective, certainly everybody has some sort of objective. Without that one, you cannot submit your project. At least in the project, you have to mention that this is my objective. Basic research. Basic research means the concept research, to understand the concept. So that is the proof of concept research one has to do. And the second one is the applied research. How can one use this knowledge which is gained uh, by doing some proof of concept research? Then pilot phase. Oh, sorry. Uh, then the pilot phase. That means scale up. Scale up is one important thing which one has to think about, because nowadays we are also facing that sort of problem in couple of products. Introductory phase, that's marketing. This is a very difficult task as far as the marketing of the product is concerned, and the final is the operational phase, that is distribution. So these are the different steps, and now one has to identify himself or herself that where at present you are fitting. Now the main challenge for researcher to enter into entrepreneurship, we say that we have such a problem and maybe one can do this. Creativity. So sometimes you have some idea that yes, I want to do something different. That is creativity. So one can start. Aesthetic, that is elegant solution for the complex problem. Just now we are thinking, talking about the same different disciplines which can come in under biotechnology. So biosensors, for example, actually sensors are being done by the electronics and other people, but when you use the bio product, bio analytic or maybe sensing unit, then we can use biosensor for early disease detection. So this is some aesthetic or uh, elegant solution for the complex problem. For example, long back I worked on the um, biosensors using polyphenol oxidase enzyme, and the Professor Hegde remembers it. We used to have discussion on this that polyphenol oxidase enzyme as a copper at the center, which is important for the activity. So he was asking me at time, I don't know whether he remembers that how to detect copper using the polyphenol oxidase. I did this one for as a dopamine sense. For the detection of dopamine using polyphenol oxidase, I worked on the biosensor. Of course, so this is the way one can try to identify the source. Then ego, yes, most of the research scientists, if you try to argue with them about their problems, etc., that ego is hurt. What is then societal benefit, of course, sometimes we think that okay, societal benefit, and then money. Just now we were talking uh, in the so Mr. Mishra said that why we start industry? We were getting money. At present, I am not getting any money because we are in the still struggling phase. So money is important to prove yourself, and this is the thing which I personally feel that everybody has to think about as a researcher that you try to prove yourself, and you cannot prove yourself by doing publication. You can prove yourself by going to the society, and that is according to the report. To give a reply to people who think research institute is a white paper, that is also true. Number of industry people think that research institute they don't do anything, and then it is just white paper. And mentorship. I I started this thing honestly for as mentorship because this my of Dr. Santosh Tukhe when he wanted to start something, I thought that maybe it will be a good idea for me to interact with him and start the company. So now we start anything about the entrepreneurship. One has to think that every concept of experiment cannot be commercially feasible in the given time frame. So one has to understand this. You cannot just say that okay, tomorrow I will start do one experiment, tomorrow you are going to start any company. It is according to me important that there is some incubation time as far as the entrepreneurship is concerned. For example, when we talk about the process scale, one cannot simply enlarge laboratory scale. You are doing some experiments in the test tube. You cannot just enlarge that to the fermenter level, 1,000 liter fermenter or 5,000 liter fermenter. It is a totally different ball. So begin with the end in mind. So one has to think about that if this experiment, if I want to make it successful, how I can go for the 5,000 liter fermenter. So that is one feature that is based on realistic biology, chemistry, or maybe engineering assumptions, etc. And one of the important things that prepare for the unexpected. And unexpected things are every day one is facing in the industry because these are the important aspects for your development. So unexpected thing is always important for your development as an individual. If you want more details, I give it because finally I'm a research scientist. So one has to do references. You can go through this paper published into the 18th, where he has clearly mentioned of the research have clearly mentioned about all the things. Now, when you think that okay, you want to start the industry, the first thing is that who is going to give you money? Friends, family, relatives, all these people, they give you money, they can try to help you, but still you need some additional support. So there are different agencies in the government. Everybody knows that if you go to it, you will find. But MSME, that is Ministry of Micro, Small, Medium Enterprises, they can give you money. BIRAC, BIRAC is now, at least the industry people are very happy that BIRAC is recognizing efforts of the industry. Otherwise, it is most of the time it is seen that the industry people are that way neglected as far as the research component or the support from the government agencies are concerned. So biotechnology ignition grant activity. Then there are two different programs where you can work with the industry. That is one is the PACE program, that is the Academic Innovation Research. that is AI program and the contract research scheme. So you can work with the industry. And I personally feel that as a initiator or as an entrepreneur, before going for the entrepreneurship, first thing that you try to work with the industry, try to have the collaboration. Then there is a group in DST also, they also support. That is a technology information forecasting and assessment council type, right? Then Nidhi Prayer, multiplier grant scheme, then scheme for promotion of innovation, rural industries, etc. So different schemes are there. And in addition to that, there are some venture capital assistance also. So these are the government agencies. You can certainly go to them and try to get something. If you have good idea, single person can also get project from the bank. Only person. If you have good idea, then they will appreciate the idea and they will say, okay, we'll give you 50 lakhs rupees. So that is a big grant. You can start. So, I'm really happy that DBT has great initiative as far as the biotechnology is concerned. So, non-government, there are three different levels. One is the venture capital. That means you are taking capital money from different agencies. Then venture center. I'm going to talk about this venture center and other is the venture studio. So there are three different levels. We can say that as far as non-government support is concerned. Now, when we take this, say that it is a venture funding. Venture funding is that people are giving money and they are you are starting your business. So that is one thing in the venture funding. There are different advantages of venture funding that you can get also sometimes valuable guidance, expertise, etc. So there are advantages, but there are disadvantages also that you dilute your ownership because that is one thing most of the research scientists they want. Research scientists don't want to dilute his ownership. Even now, I feel sometimes that if I go with somebody, then certainly it is I am diluting my efforts for doing that sort of research or that sort of innovation. So this is venture capital. Now, venture center. That is one thing which actually there was one lecture. There could have been one lecture in the afternoon from the venture center. But somehow I am sharing my experience and I am not very happy with the this concept. When we talk about venture center, usually we uh, expect that that startup is supported by venture center. But no, I find that the venture center is just running with you. You are yourself driving the cycle, learning the cycle. And third thing is that which somehow I experienced that one that business development and venture center, you are running your cycle, you are driving the cycle, and they are just taking the advantage of your efforts. But venture studio is one of the important concepts which you can think about this. That when you cannot, you don't need capital per se, but you need idea and the help, or the somebody should be there always with you. So that is the thing which venture studio they can give. That venture studio does not fund existing startups, but they can certainly uh, give the idea that okay, this is the way you can go, and we are there with you. So if that sort of strong support is also necessary, and eventually this venture studio employees can decide to stay in the portfolio of the company, or they can go away from your company and they can start new venture. So that way, this concept, according to me, is certainly interesting and can help the research scientists who are entering as an entrepreneur in the in their desired field. Again, we need some internal company analysis and market. The industry person one has to think about these two aspects. As far as the market potential is concerned, that is your product or service unique. Of course, at this point of time, I don't think that you need all these things. You need the startup. That yes, your idea is industrialized or is could be commercialized. So that sort of thing is necessary. But These are the points which are important. Is there a demand for your product or process in the market or even service? What has been the industry trend in your sector of business? And has the industry been growing and what is the outlook for the future? So these are the things which one has to slowly think about if you want to go for the entrepreneurship. Now, there are two different aspects. Whenever we think about that starting the company, even I also thought about that one, that we should have some advisors. So advisors are not at all useful. You need some mentor with you, and that mentorship, according to me, is important. Mentor and not advisor for young entrepreneurs, because mentor, even if it is necessary, can give money also. So because that mentor always thinks that this is my company, advisor thinks that I am advising the company. So these are the two different aspects. So it is necessary. Now at present, what we as a mentor, what I have done that to provide platform for translational research in agriculture biotechnology. So it is not only my company. I am just trying to do some service also to the people. If you have any good idea, if you have any good proof of concept, which can be scaled up and then which can go for them in the market, then certainly you come with us on this platform with necessary MOU, MTA, etc. And then eventually you can, whenever you are ready, like it is some sort of venture studio type that we have started. So, and we are actually helping different people also to different industries. Now, why I started this company? To prove myself and eventually see the world through young eyes, because then this is the win-win situation. Because my partner is young, I am maybe at the fag end, so at least this is a win-win situation for both of us because success is important. So what is the role of mentor? He can motivate team because he has a lot of experience, maturity. Then help, we can help to focus, skill training, and flag risk. This is important. Mentor is always there. Advisor will not tell you that okay, this is the risk in such and such process or product. Advisor will not tell, but mentor will tell because mentor is emotionally attached with your company. So according to me, mentor is important, not the advisor. And candid feedback is always there from the mentor. But okay, you are interested in technology, if you are interested in company developing, but still you are not very comfortable in raising funds, etc., etc. So maybe this is the way you can have incubation center, which can create facility. Then you can give the facility, rent the facility to others. For example, culture collection, you have different instruments, and then you can rent this instrument. But there are no intellectual input and this is not creativity. Then one-stop shop. Just now I mentioned about the platform for the translational research, and that is the idea which I was in the one of the PBT committees. That time we decided that we should have transfer platform for translational research on biocontrol because to promote organic farming. And I got the support from PBT, but eventually that money was not sufficient. Anyway, that is different. So this is creative and challenging and do it yourself technology that is most challenging, that you are now doing only social service. You are developing industry and eventually you will enter into the social service. So this is finally do it yourself technology. So now, which is your true now? Try to identify there are two different areas. Healthcare and pharmaceuticals is one which is high value, low volume area. Second is agriculture or wastewater treatment and etc. Others, which is high volume, low cost. So these are the two different areas. I am rather dividing total say industry sector in these two areas. So what is the importance when we talk about healthcare pharmaceutical? It is individualistic, while agriculture is community based. So now you have to think which way you have to go. I'll just give one funny comment in the last. Uh, Success is defined as far as the healthcare pharmaceutical is concerned. If your proof of concept is good, then certain success is defined. Here, multiple factors are involved. And about these multiple factors, uh, I can tell you that one funny incident. I wanted to work on the chitin synthase enzyme for the screening of chitin synthase inhibitors. And one pharmaceutical company approached to me, and I thought that maybe I should work on this production of the purification characterization of this enzyme. Then, uh, then our HOD, I can tell his name, Dr. H.S. Srinivasan, because of him I entered into agriculture. He said that Mukund, you should not work. I mean, don't take it in other ways. It is just joke. You should not work in healthcare. You should work in agriculture. I said, why? If you are successful, success is yours in agriculture. And if you are unsuccessful, you can blame it on climate, heavy rain. And then that's why I entered in agriculture. I mean, joke. No? So this is the thing which is important. So here multiple factors is involved. And here it is, there are no multiple factors. Of course, I mean, it is a generalized statement. Um, it needs almost 12 to 15 years to get the one product approved. And here it is 10 to 12 years. So time frame is almost same. So what are the steps? Proof of concept research in both the cases, preclinical trials, clinical trials, and FDA approval, etc. And maybe Professor Lushra can highlight more about that one. But in agriculture, same thing, proof of concept, scientific field trials with agriculture university, then demonstration trial in the large scale, then you go for farmers participatory trial, and then maybe central insecticide board registration or the state approval, etc. So the states are in both the cases same, but this is a high volume, low cost, and this is a high value, low volume area. So now you have to decide which way you want to go. Don't go by that joke, but I personally feel that healthcare pharmaceutical is more challenging area as far as the application is as far as the product evaluation is concerned. In case of agriculture, though I am working in agriculture, I personally feel it is not that challenging. So opportunities in biotechnology, research and development to entrepreneurship. So now, if you decide that okay, you want to go for entrepreneurship, the first thing is that what are you going to do? So there are different problems. Say you can work with bacteria, you can work with yeast or fungal organisms or mammalian cells, plant cells, animal cells. Then you have to identify yourself that uh, what are the relative merits for different life forms. Speed, what is the growth, how that organism is grown in the laboratory or in the for that matter in the fermenter. Costing to grow that organism. Then post-transcriptional modification. This is one thing which is important as far as the eukaryotic systems are concerned, which is not there in prokaryote. They scale up, and then the regulatory aspects. The regulatory aspects means not the registrations, etc. Regulation of the production. For example, glycosylated proteins are there. If you are producing the glycosylation, one thing, or maybe lipid molecule moiety is added. So all these things. So if we think about these aspects as a one has to think about all these aspects, you will find that the east and kilometers Punjab are always in the better position. So one can start not because I am working on the Punjab, not because I am working on the east like say, but uh, they have these better characteristics. You will find as far as these industrially important or commercially important aspects are concerned. Now, I work with microorganisms. So, what is the advantage of working with microorganisms? Just I thought that maybe this will highlight you. One industrial microbiologist, Professor Perlman, he said that the microorganisms always like their trained and sensitive partner. There are no stupid microorganisms. Stupids are human beings. Microorganisms can and will do anything. Microorganisms are smarter, wiser, more innovative than genius, engineers, and others. If you take care of your microbial friend, they will take care of your future. And that at present I am experiencing. We have different products using microorganisms, bacteria, as well as the fungi. James Watson, everybody knows molecular. I talk about molecular. I mean, I have specifically put this quotation quote because molecular biology is there.
If we can solve the same problem equally well with ease or with human sense, common sense tells us to stick with the simpler and less expensive system. So that is one thing. Of course, I mean, I don't want to criticize any research which is going on in other research. Third one, in that introduction, it was mentioned that I organized one microbiology conference, and for that conference, the chief guest was Professor Ramana. This he was chief guest. He is physical chemist, though he was doing some microbiology at that time. And he said that I don't understand microbiology. So I thought that maybe in my conclusion, I should say something about microorganisms. So I said that I'm not as big as these two people, but the perfection of microbes far exceeds the accuracy of physical chemistry that is beyond our comprehension. According to me, this is one of the important things, and that we are going to do microorganisms. Now, couple of examples, quick examples, healthcare, architecture for agriculture. News in newspaper has life of 24 hours. So when somebody from the research institute says and something is published in newspaper, I ignore that one. It is not a starting point for start. So one has to think about that instead of going to newspaper, one has to have the good scientific publication. Go for the publication, go for the patent, go for the technology transfer. Then I can say, and then eventually you can give the interview to the newspaper. But most of the thing I find it the other way around. By doing just by just doing one essay in the plate, say antifungal essay or antimicrobial essay, if you give the interview in the newspaper, then I certainly feel that that person doesn't have any future as far as the some translation activities concerned. Hotspots like Western Ghat and Antarctica have not given any commercial antibiotics. I mean, they must have given some products, but not any. So, but everybody talks about Western Ghats. I mean, I don't want to name, but recently there was an interview of one scientist in the newspaper that he is screening different organisms from the Western Ghats and trying to get some antibiotics which will uh, kill the resistant pathogens which are developed in the human body. But unfortunate part, the irony of the situation is that screening using laboratory strain has no meaning. You are doing screening using laboratory strain, and on the other side, you are saying that you are trying to screen the organisms which are trying to develop antibiotics for the organisms which are resistant to different things. During COVID time, everybody was talking about mucormycosis. And mucormycosis, people have even said that it is because of penicillium. Actually, penicillium is not a mucor, but number of people they try to write something on the antifungal compound using plate essay with laboratory grown isolate, which has no meaning. But their research and everything that after the COVID was over, everything was stopped. So this is the important aspect as far as the what should I say the path. Going towards the entrepreneurship. Don't go to the newspaper. You can go to the newspaper when you are successful. I'm just giving little information about the because I worked on this aspect. Though Dr. Srinivasan told me that I should work in agriculture, I worked with different companies, even ex pharmaceuticals also on the antifungal drug development. That rational for selecting antifungal. And my favorite area was the cell wall of the fungal organism is the novel target as far as the specific antifungal compounds are concerned. So target protein should be present in several fungi. So that was my first aspect. And it should be present in fungus. It should not be present in other plants and animals. For example, target for chitin synthase enzyme is chitin, and the chitin is absent in human being as well as plants. Though chitin is enzyme because it is necessary for the plant as well as the human beings to control the fungal infections in their body. Chitin per se or chitin synthase per se is absent in the plant and human being. So that target can be a specific target, and the protein should be essential for the growth and the virulence of the fungal organism. So we identified this writing synthesis. And of course, one can use bioinformatics approaches. Another approach one can derivatize the existing uh, uh, known inhibitors for the better antifungal agents, and maybe you can go for the type. This is another novel idea. I can say that though I have not worked more on the extensively on this, dimeric or multimeric molecules can be prepared, or hybrid or dual inhibitors can be prepared, which are having the broad spectrum. So this is the development of say antifungal agents, and now you will find that the number of I was thinking about the nicomycin like molecules, which is at present in the phase 2 clinical trial. So like that, these are the development of antifungal compound. So one can really think about the development of such type of molecules eventually in agriculture. Now again, this is our government has some certain vision, and they want to make a 5 trillion economy by 2024. Actually, it was 2023. Now I changed to 2024, but still today we have not gone to that. About 50% of Indian population has agriculture as the primary source of livelihood. So I thought that maybe this is a good idea to work with the agriculture products. Farmers are facing problems of uncertainty in climatic conditions. So naturally, one has to think about agriculture as a major sector or major area which can be used, which can help to India to make a 5 trillion economy by 2024-25, etc. So why and how the change from rural livelihood sector to modern business establishment. So can we change this rule because farmers they are having small land and they are producing something and then they are eating themselves for the it is good for only family. Uh, so real-time advice to the growers, digital marketing, avoid unorganized sectors which produce spurious products, etc. That can help to reach to this target. And the technology like nanotechnology, so far it is not successful except nano urea, but that is also not successful. Hydroponics, growing plants without soil. And just now we were talking about chemical fertilizers. So in the soil, what will happen to the growth one? Vertical farming instead of because land is less, so can we go for vertical farming or use of artificial intelligence in agriculture? And number of people are doing, and number of computer people or information people they are now entering into agriculture. So these are the different avenues we can say. Uh, The setup in agriculture certainly change. This can change the scenario because lot many farmers they have committed suicide and because of the family problem, farming related issues. And this is, I think, recently there was some problem about the poor quality farm seeds, the lack of education on best practices in farming. Because that at present we are doing this one also. We have started our own WhatsApp group where we are not promoting products. There we are just discussing problems of the farmers. And now at present we have 500 farmers on that one. Every day we get information, questions from them and Santosh because he is very good in interacting with farming community. So he answered them. Yeah. So these are the different priority areas in agriculture or commercial ventures. Agri-clinics where you can have endophytes or as bodyguards, and that is the organisms which are present in the plant, which can survive in the plant symbiotically or mutualistically, can eventually become bodyguards of the crop. And I just talk little about this one in the later. Disease suppressant. Instead of treating disease, can we have the soils which are disease suppressive? And this is the novel concept at present one should think about this. Nano fertilizer, nano biopesticides, and nano biopesticides okay, but nano fertilizers which has a lot of limitations. Hydroponics, vertical farming, biodiesel production, then all composting, food processing, hybrid seed, all the other areas, or even last is the vaccine production against plant pathogens, which is not there. Vaccine production. Only in 1931, somebody tried to produce a vaccine for the against potato disease, that is phytophthora, but nothing has happened afterwards. So these are the different areas in agriculture which one can think about going for the entrepreneurship or the next venture. But one has to think about the two important aspects which are necessary. If you want to start any company, this like according to me is important thing. Other things you can certainly manage. Socio-economic issues and challenges. These are the unfortunately mathematics for marketing. It is not agriculture produce, any produce. Manufacturer, consultant, distributor, shopkeeper, and farmer. So manufacturer has to give very take only low had low price for the product, but farmer has to pay higher price. Same thing is true in pharmaceuticals also manufacturer because this distribution line is taking all the benefits. Farmer, broker, market, and the customer. Or uh, mm, here also you will find that this in between people and at present you must have read about the tomato and onion etc. Farming communities in always issue. So they are having all this problem. So this is these people they decide everything. Here also these people they decide everything. So if you start your own entrepreneurship, at present we are facing this problem. Somehow we are trying to solve this problem. But this is according to me is one of the important slides I can say in the starting any entrepreneurship work. So now I'll just talk about my work, how why I started and why how I started this. It is organic farming or the eco-friendly agriculture. And here my thinking was that apply science for the good of the people and traditional based knowledge based versus knowledge based products. And I am fought for this knowledge based product and not the traditional. That again, don't quote me, but that gomutra and share and all sorts of things. Somehow I am not very happy with that. So I am happy with the knowledge based product. So now this is the potential we can say of any agriculture product. Just now we were discussing about the organic farming. Whether organic farming is in India, if you ask me, not that much. Only 5% organic farming is there. Real organic farming. Otherwise, there is no organic farming because in India we use agrochemicals around 0.58 kilograms per hectare. In USA, they use more, 4.5 kilogram per hectare, Japan 11 kilogram per hectare, but our consumption is big. Chemical pesticide market is around 66 billion and biopesticide market is around 6.6 billion. So what is the scope for your biopesticide market is around is 10 times more. So you can bridge this gap and maybe eventually you can go for the organic farming. Yeah. So what I did, that as I mentioned, that the microorganisms are important um, for any entrepreneurship. So in this case, there are I worked on the microorganisms. There are fungi, bacteria, viruses. Then one can have the now it is a wide spectrum where we can use different enzymes produced by the microorganisms per se, which can degrade protective cover of insects and pathogens. This is one of our novel products also. And to increase the efficiency, maybe one can have the enzyme inhibitors, which can control the virulence of the organism, etc. So these are the three different areas which we have identified for the product development. So now this is the journey from laboratory to farmer or the farming farmland, you can say. When one is always proof of concept research. Whatever you are doing, you try to have the good proof of concept. And proof of concept doesn't mean that only one experiment. And it is necessary to repeat your experiments and try to do the as much concrete proof of concept as possible. Second one is of course the demonstration of proof of concept because that is another. area, which is important, and the farmers participation program, where you can convince the farmers and then try to see that yes, your work, your product has some potential, and eventually transfer of microinsecticide technology to the private sector, and then finally regulatory considerations like central insecticide board, etc. So these are the different steps I have already mentioned about this one. But one more step, which is according to me is important, is the new age, the bioprospecting and value addition. You should not stop only at one stage. You have to think about that how you can reduce down the cost, because industry its cost reduction is one of the important thing. Either you can have the value addition or you can reduce the cost. So this and without affecting the quality, that is according to is important. Certainly, there are certain products in the market that they are very cheap, but quality is not there. So one should not think. One should always think that quality is important. So try to go with quality product. Innovations in applications. I mean, this is just case study. So, for example, while producing this infective, that is the spores of organism like metanism, we have used some membrane bags where the heat exchange is there, gaseous exchange is there. So you can develop the good spore mass. So that is one thing which we have innovated during our production program. Then there are different sprayers also. You will ultra low volume sprayer instead of regular sprayer, that is knapsack sprayer where the volume is high. Here for per hectare, usually you need 500 liters of water, while here you need 50 liters of water. So this is the way you can save water. And then again, the important aspect is that you should educate farming community, because that is one thing which is lacking. And so at present we are doing that well also. So now, why I started all these things? Because actually I was highly interested in biodiversity part. That was my main idea. That why we should go for the such type of venture using the fungal organisms in the field. If you grow your organism, it is a insect larva, and you will find that the fungal spores are produced on this larva, day larva. Usually it is in 7 days, but if you do the regular subculturing of the organism in the artificial media, you will find that the virulence goes down. So, and it is true for all other organisms also. Any pathogen, virulence will go down because if you maintain the organism on the artificial media. So naturally, we found that the protease of culture is having the low virulence. So naturally, it takes 12 days for the sporulation. Here it is 7 days. So then that means repeated subculturing should be avoided. And then if you want to regain the virulence, then you have to transfer that organism on the insect, that insect passage, early insect passage, you will get the virulence. So then I thought that maybe instead of just having the repeated subculturing on the artificial media, can we grow this organism along with the insect pest, along with the crop at one place? And that was the idea to develop biodiversity park. And DBT principally approved, but again, when it comes to money matters, and somehow it did not work. So that was the main idea of working on the organisms uh, which are being used in the field. Everything started in my room number 1871. I am still quite passionate. So we had two patents, many publications, etc. And we had four different products which we have developed: the meta-rhizome anisoplate, cuticle degrading enzyme complex, or the micronutrient enzyme complex, a fungal chitosan, and that is useful in healthcare, and then essential oils plus chitosan-based fungicide. So these are the different products with which I thought that maybe we can develop the technology, we can go to the field per se. Then we submitted one application to DBT big grant, but unfortunately DBT big grant has the healthcare as a priority and not agriculture as a priority. 3 times we presented there, and some of as agriculture is last priority, and there were no fancy claims, and that is one thing which is important thing that in when we talk about biotechnology, everybody thinks that recombinant DNA technology. Recombinant DNA technology has not really seen the light of commercialization, except in the production of extracellular enzymes, etc. Otherwise, in agriculture, recombinant technology is not acceptable for various reasons, and you must be seeing that even the genetically engineered crops are not acceptable because number of geopolitical issues are there. So, but and whatever technologies we are doing, it is the classical biotechnology. Just now we were having breakfast about the idli, and then that time the when I was working in say a long time, this Professor G.D. Gulab, who was editor, chief editor of biotechnology latest, and that time I organized one workshop in Mahabaleshwar. It is a recent trends in biotechnology, and everybody was talking about molecular biology, including our director, sir. So everybody talked about molecular biology. Then J.D. Gulab he, he said that whatever you are doing is not biotechnology. Biotechnology is leaf fermentation. That is one of the important things what he said. This is the classical fermentation. That is classical biotechnology which has reached the people. And that is according to him important. He said he used to publish those papers in biotechnology latest. It is one of the good biotechnology journals. So anyway, so there, this was the story. Then we thought that maybe we should have our own company. Then we license all the technologies from NCS officially. So all the cultures, etc. we took and then started our own company. So what are the weaknesses? This is one thing which one has to do when we start any company. SWOT analysis. What are the strengths, what are the weaknesses, opportunities and threats. And without that one should not proceed further when you talk to your company. Like, for example, in case of our micro insecticide, like I say, it is scientifically developed process, high efficiency, low treatment cost, etc. I mean, okay, I have not gone into details. Weaknesses, end use market has less product per quality perception because farmers usually think that okay, when they say pest in the field, they spray chemical and within seconds chemicals also die. Within this insect die, and eventually they don't understand even human beings also will be affected. But now farmers education is necessary. That is a weakness. Opportunity. It is a growing market. As I mentioned that at present biopesticide market is 6.6 billion. You can go up to 66 billion. It is a total pesticide market. Threats. Know-how cannot be kept confidential because we are using living organisms. So any microvirus can isolate the organism and can and unorganized sectors are there. And the main hurdle is of course the central insecticide board registration. So that is main, and we are not talking about other aspects as far as the government organizations are concerned. Now these are the issues and challenges. Okay, you have one product. Now what are the different issues and challenges? Virulence of the organism. Just now I mentioned that you cannot maintain that organism in the artificial media. Speed of the action because farmers need fast action, large scale production, etc. And from industry point of view, cost and IPR, that is the intellectual property rights. Now, these are the different steps one has to think about. Product is evaluated for a single pest. Microinsecticide we evaluated for one insect pest, that is Helicoverpa in pulses. Then next step is the protection of IP. Then third step is the maintenance of effectiveness of the spray. Then identification of the backup strains. Then next is the product evaluation for multiple pests and pathogens to reduce the cost. Bioprospecting for validation, and then novel approach formulation with multiple strain species. So at present we are here, and now we have to go up to that those points. So when we talk about protection of IP, live organism we cannot patent. Native organism only recombinant organisms can be patented. So naturally, our organism is the natural isolate, so we cannot pattern. But we can pattern something which is different. These are the microsatellite marks, and microsatellites are strain specific. And these are though these are from the non-coding region, these are specific for each strain. And using these microsatellite markers, which you can patent and you can identify your organism. If somebody takes your organism, certainly you can challenge that one using your microsatellite markers. So that is for the IP protection. Maintenance of the effective strains, that we can have the insect facade which can where you can regain the virulence. But is there any other way to maintain the quality? And that we have developed on our own. That what is the new aspect? That it is that the puzzle of highly virulent metarhizium manuscript strain from Annona, that is uh, against the Helicobacter armydia. What we have done, we have isolated almost 68 strains, and okay, I'll just go to it. So these are the different strains we isolated from different crops: tomato, custard apple, sugarcane, brinjal, okra, pigeon, etc. And then we found that the isolates from this custard apple field, they have some important property, or rather they are more effective in the field. And what could be the reason for that one, that we just wanted to find out. And we found out that there are these mechanism isolates from the custard apple field. They were virulent, and then we changed the custard field. We collected a soil sample from different custard apple fields from different Maharashtra areas, and we found that these organisms are more virulent, which are coming from the custard apple. So we realized that what could be the reason, and we found that there is a horizontal gene transfer from custard apple to the to metarhizium anisopli, because custard apple is having a lot of insecticidal properties, and naturally you can see that insecticidal traits from custard apple. They have come to the metarhizium anisopli, and all metarhizium isolates from the custard apple field, they were more virulent as compared to the isolate which are from different crops. Then we validated this concept that okay, chili is also having different insecticidal properties, neem is also having insecticidal properties, and papaya. We isolated metarhizium strains from those ones, and we found that okay, they are having more insecticidal traits. So now instead of going for the insecticide, maybe you can transfer your organisms into the custard apples, or and develop it as an endophyte, and you can get the better virulent strain. So next is the selection of commercial strain and backup strains, because you cannot just have only one strain, and if that goes, if it is contaminated, if your microbiome is not good, then you cannot do anything. So instead of that, you should have some backup strains. So backup plan is always important. Rather, you first develop backup plan and then go for the commercial. Without backup plan, one should not go for the commercial use. So these are the backup strains, two backup strains, this one and this. And then we have characterized them, and then we are at present we are maintaining these strains. Rather, at present we are maintaining 10 different strains, which are having the first line, second line, and third line, like that. So then multiple hosts. So then we tested organisms again different ways, and we found that the cost can be reduced, because the one organism can be used for the control of different insect pests as well as maybe pathogens also. And then this is according to the important for the cost reduction. Then bioprospecting. After cost reduction, you can do value addition also. And here you can have the use of this material and supply for different things. You can have the broad spectrum against pathogens, pest and pathogens, for example, pathogens like fusarium, etc. Then these organisms produces different enzymes. It produces lipase, protease, chitinase, etc. And lipases are useful. This is the extracellular enzyme. So when you grow your organism, conidia you can use for the control of pest, and the extracellular growth you can use for the isolation of the lipase enzyme. And which has lot of applications. Same thing for protein, which is being used in detergent industry. Even you can use this organism for the bioremediation because it is not that all agriculture farming is only organic farming. People are using different chemicals, and in that case, you can certainly have the organism which can degrade also these chemicals. So this mechanism can degrade the chemicals and bioremediation of pesticidal residues can be done. Or you can have the nano factories bionic. I mean, this is just a piece. I can say that the laboratory experiment at present we are not done very much on this one, but you can use meta-resin mycelium for the synthesis of silver nanoparticles. That one we have seen. And now the silver nanoparticles along with the meta-resin you can use in the field to control different pathogens. You can also isolate the cell wall after producing this conidia. The remaining microbiome you can use for the isolation of chitin chitosan. This is the polymer of the fungal cell wall, and chitosan has lot of health applications. For example, to reduce down the obesity, you can get the chitosan tablets. For the arthritis, you can have the N-acetyl glucosamine or the glucosamine for oligomers for the for that arthritis problem. So like that, healthcare applications are also there, and you can have different enzymes along with the metabolism and so like chitin deacetyl enzyme. I mean, it is too technical, so maybe we'll just um, skip that. Important point is of course the education to the farming community, and even we have produced one technology film also to reach to the society, and that is according to important because product, I mean, unfortunately farming community or for that matter the end user doesn't have any perception of the quality of the product. So instead of that thing, if you give the information always to them, so certainly it will be useful for the farming community. So novel approach formulation with multiple strain species and genera. So it is not only meta-rhizome, you can mix meta-rhizome different strains, you can different species and genera, and we can get the effectiveness. This is because every strain or every genus or every species has different type of mode of action. So naturally, multiple organisms can certainly work better in the field. At present, we are doing the same thing. Then you cannot just stop with one product. You should have something in the pipeline where you can, otherwise your industry will not survive. So we have one more product that it is Myrogold, that is the enzyme preparation, which, because once in DBT we had discussion that we should have wide spectrum biocontrol agent, which can control. That means only one spray, one type of spray, which can control both insect pest and pathogens. So we have developed one enzyme formulation from one organism that is the uh, myropatium. And from that one, this organism produces enzymes which can degrade protective cover of insect, that is the insect cuticle, and fungal cell wall, which is again the protective cover of the fungus. So we are trying to develop this product, and then maybe in another 2 or 3 years we will be able to bring out this. And we are working on the nano formulation, enzyme nano formulation uh, of this enzyme preparation. So again, this enzyme preparation we are tested in the field with the farming community, and it is controlling different types of pests and pathogens. And nano formulation also we have made along with other Research Institute, and we found that the lipase and endochitinase, these are the important enzymes which are entrapped in the alginate as well as in chitosan base, and are up to being tested in the field, but not at commercial level. Products at scale up phase. So this is one aspect that okay, this is the product scale up phase is over. So that is second step. Third step is the product at scale up phase or mushroom cultivation. That is important. And why mushroom? I'll let you know. And chitosan. So mushroom has cell wall of chitin chitosan, so we can isolate these. So these are the two products at present at scale up stage. These are some different mushrooms which we are growing in the laboratory. Fungal chitosan and we'll go with next step. Uh, and product at concept level, the proof of concept level, the biocomposite. At present, people are interested in the developing biocomposites. Usually, we use cellulose board. Instead of cellulose board, can we use chitin cyclosan board? It is a fungal biomass. And what they do, we have, for example, we take the straw and we grow our organism, that is mushrooms or any organism, which is producing filaments. And that filaments, they are holding the all the mushroom straw together, and then you can make the board. And as the chitin is more stronger than cellulose, these boards are important. Even mm, uh, this is the biocomposite, and you can have different modules for that one, and then you can uh, make this biocomposite very effective. So at present, we are also working on this type of aspect. And the probiotics for poultry, that is another. So we started with proof of concept for two products. Presently, we have 10 plus products, and products registered with CIB, etc. are also there with us. In addition to that one, we are DSI recognized. So because R&D, and please don't forget that though you become entrepreneur, you should not forget your roots. You are research scientist. So research is always important, and that is according to me, one has to think about always. So at present we are having different R&D projects also, direct projects as well as. So at present we have one project with Assam Agricultural University, then with the so like that R&D projects are going on. Maybe eventually we will come up with different products. Be prepared for the unforeseen problem. I we started in November 2019, 3rd November 2019, and after 2 months everything was stopped. For 2 years we could not do anything. But that time also we started working. We were doing some work. We could I could publish one book in COVID, and also this to tell people that mucormycosis. What is mucormycosis? So all these articles that time I could write, and this is the these are some publications for the especially for the agriculture community, and usually I write in the newspaper for this. In addition, this is my passion, mycology and microbiology and postal stamps. So these are the different articles I wrote, and I'm writing in the for the popularization of our concept. Now, this is my one of the recent articles. There is a big gap between scientists and industry. What is the reason? Transfer of technology from the research institute of university is a major concern because you work in the research institute, but you always think that okay, industry is just trying to get everything from you. So it is not true. And that's why what is happening, though the scientist is modest, the scientist always is modest individually, but business development group and higher authorities demand unreasonable amount. Because when I go for some product, etc., I know the what could be the level it can reach up to. So in that case, I will not for 1 lakh product, 1 lakh unit product, I cannot give 10 lakh rupees, but they don't understand and they try to demand. And the unfortunate aspect is all others get handsome share except the project scientist. So that is unfortunate situation in most of the cases. So try to see that this gap is, and always we are sitting like this back to back. Instead of back to back, if you sit like this, certainly industry academia interaction will be useful, and maybe this can give me pleasure that whatever I had discussed with you, maybe you have also come out with a good entrepreneurship. These are my recent publications during COVID time. These are the 4 books, and in this book we have mentioned about the biocomposite, fungal biopolymers. Well, thank you so much for your kind attention.
Thank you so much, sir, for this inspirational talk. If there are any questions, we can take.
Has to be some questions for today because we have
I think there is more opportunity in doing business and something than just taking another job somewhere. You should think about going into this kind of thing. But this whole idea of organizing this is to inspire you, encourage you to take up your own entrepreneurship. So please think about it. When you take this, you will empower people. So please think and get so get because I think many products here, many of them, and it is mentioned venture studio. I very much thankful to you. But I think that is the way I will myself think of doing in that fashion because whatever products we are developing, I think I want to play and then think, encourage people to take up that product. But this has been, this journey has been very encouraging. People should think now you are scientist, he was scientist, now he is the industrialist. So please think, please ask questions because that is necessary to improve my skill. To improve this.
Thank you very much for the nice presentation. I'm Harshad Patil. I'm a scientist here at IRSHA. So I agree with you that at Venture Center, initially we get the good support, but later on they try to extract more from us. So therefore, I want to know what is the could you please elaborate more on the concept of venture studio?
Yeah.
And second, definitely we can write, we can get big or so that's it. But that amount we get for the industry is quite limited for a startup. And thus we approach these people, investors, who can give money to us. But then dilution of the shares is a big factor. So how much should we dilute our first share? And if you think
Maybe question. Okay, first question is about the venture studio. So if you ask me, venture studio is nothing but a concept. Even I also started off entrepreneurial research because everybody cannot start any industry. So I have my facilities, microbiology facilities, biotechnology, field performance training, etc. If one has any concept, that person can hold hand with us and then we can go ahead and then that person can go out with his own idea or maybe he wants to continue, then we can continue. Same concept is there in venture studio. They are there to share their knowledge because it is not that the venture studio is tailor-made for you. Venture studio has different scientists having different capabilities and they develop different projects. Now, if you are interested in taking up their project, you can certainly take up their project and you can start your own company on that. And the venture studio will be always be there. They will work with you. They will take you to the certain stage. And if you think that okay, now their role is over, they can go out. They can have their different ventures. So, like that, venture studio is not the liability. This is one thing. Venture center now maybe we can talk about more on venture center. Venture center. So why initially I also wanted to go to venture center. I started with venture center, but I realized that they said that our share will be not only for this product. Our share will be in the company, and our one person will be one of the directors. If their person is becoming director, that means I am all the time I am just giving money unnecessary for my other activities also. So I worked in venture center for one product and they wanted to claim for certain products. So I thought that maybe venture center is not right. But studio is not that way. Venture studio is yes. So in that case, venture studio, I find that they will work for you. And of course, certainly when they are working, they will get the benefit of that also. But they will go out. And that is according to the good board. So venture studio, if you ask me, venture capital, that is sharing your benefits instead of with venture studio, venture. So that is what I feel. Your second question was,
What is the dilution of the shares?
the dilution of the share in that, of course, that is in venture capital certainly, because I have made mention about the disadvantage of the venture capital, that dilution of the your ownership, that is there when we say of the venture capital. So that disadvantage is
But how, to what extent we should dilute our shares?
to what extent you can. It is very difficult because if you take their support and from that support, how long to think about that, and without taking their support, how much you can go. If you think and you compare both, then you can decide the share.
I think you are said the thing would will be able to do it.
Many.
So very well. Okay, Chitra. Let me understand what the question was from each segment.
Then I mentioned three aspects. One is the venture capital, the studio.
Venture capital. Venture center and venture studio are two incubators.
Not incubator. Incubator you need. Venture studio you can.
Venture center. I will clarify.
For the TCE or to then for
That's
the
the reverse.
I asked Asha.
So then very well about venture centers.
I worked in a venture center initially. I started in venture center with my big grant. But then I realized when they said that, okay, our one of the persons will be director of your company. No,
PIG.
let me complete. And the second thing is that that director will have share in all other your products. It is not only one product which is no, I may be wrong or not wrong. That person may be wrong who has given this. And then that time we decided that instead of going through venture center, we should have independent company.
We can discuss this again separately.
Maybe. No, I mean now ball is in his court, so we don't know. No, it's not. I'm not saying that this is good or this is bad. Anyway, nothing personal. It is. It is problem. When they said that okay, our share will be there in all. Yeah. Then I said that we are working and we will do the most.
Exactly. Okay.
No, he will carry.
Maybe you can.
Maybe they are responding. Time is there, na? Can I talk something? People may be knowing me, but after leaving the IRSHA, what I am working now, coincidentally, it is related with Dr. Mukund. Actually, let me tell Dr. Mukund and we are very close friends. Maybe from 76, 75, we are together. But his journey is really interesting and very outstanding. My purpose of standing here that at present I am doing some consultancy where I am using this enzyme lipase and I am working with Novozyme enzyme and Fermenta enzyme. Just now I found that if Dr. Mukund can spare me some lipase as well as chetosan,
then probably we can work in collaboration with his company. It will be beneficial. Only thing is that I am not the owner. I am only a consultant. Actually, he is a consultant and are good people. Unfortunately, I am doing consulting. Yes, I am doing the big department for green chemistry project. The company is already established something of more than 1,000 crore turnover. They have their established product like fatty acid esters. At present, they are developing their fatty acid esters with green chemistry. They have already a big plant with big plants with 10 KL reactor vessel. At small scale, I am doing the enzyme lipase. And our primary work is going on. When I saw that Dr. Mukund is already producing lipase and chatosan, I think it's a good opportunity to work together where we are already a old friend from almost more than from 76. Thank you.
Anyone? Okay. So we'll wait for tea right now for 10 minutes. So, but I wanted to announce one thing that since we have all the speakers here now, we will do the inauguration ceremony after the tea break. So in 10 minutes we meet again. So it's 7:45. So 11:55 we'll be back. Okay?
Just suddenly picture Palestine, right? So,
पुढे घे हे पुढे घे अजून पुढे पुढे घे हे किती पुढे पुढे घे हा पाठवलं स्पेस आहे दुसरं एक मला समजत नाही तर स्पीकर बोलताना नाही नाही हे लिहिलं पाहिजे एक बॅनर करा आणि ते सांगते बरोबर येतं बरोबर पण आणि काय सर आम्हाला एक जाणवतं मी वर पाहताना शिस्त सगळं ब्लँक दिसते ब्लँक दिसते थोडं पाठवा बस बस आता तुमच्या नाही नाही